thank you so much to our speakers to have given us a good understanding of how innovative technique toward extrapolation and efficient drug development can be used. So clearly a big thank, and also the opportunity to welcome them in the panel discussion that, I'm pleased to moderate together with my colleague, Lin Yao. And it's a great honor and pleasure, Lin, to have you with us moderating this session. When I'm saying with us, it's also together with my colleague and friend, Amy Cheung. So it's, I'm sure there are lots of questions already, knowing that we all have also as moderators, lots of questions, knowing of course the experience and the expertise of the colleagues and knowing, what they have shared with us. So I'm sure that we can start with maybe you, Lin. You have a first question to start with.
I would, and I do want to thank Dr. Batchelor, Dr. Nelson for really terrific talks. I'm going to ask a question to both of the presenters, and I'd like to hear your perspectives on this. And maybe we'll start with Skip first, and then I'll ask the same question. So the question is, in terms of model informed or modeling simulation, Skip, in your opinion, in your experience, where do you believe the highest impact areas are for use of this tool in an extrapolation approach? And I'll ask the same question to Dr. Batchelor, but you mentioned modeling and simulation as a way to evaluate these potential differences in bioequivalence between adult and pediatric populations. And I would be curious to hear on your end, again, which approaches or what strategies might be most useful, and in which populations this approach might be most likely to identify differences. So I'll start with you, Skip, and then Dr. Batchelor.
Well, Lynn, thank you for that simple question. So I'll just make some general remarks. I think it's the challenges are different for small molecules and large molecules or biologics, and at least I don't do the modeling, but as I'm a consumer of the information that those who do modeling provide to me, my impression is that the amount of data we have to inform, let's say, physiologically based PK model development is much more robust in the small molecule space. And so that may be a bit more mature. In biologics, for example, there's more challenges in modeling the doses in the lighter, younger kids. There appears to be a greater clearance, not clear why, hasn't been worked out fully relative to things like the small molecules with the cytochrome P450, et cetera. So if I had to say where there are the challenges are in those lighter, younger kids where there are some changes that might be going on, both, in terms of the maturity of ADME is generally under 2 years of age, but even the prediction of biologics for kids that are less than, say, 30 or 40 kilos is a little bit more challenging. Now, I say that with the recognition that often that's the very same population that's the hardest to enroll in clinical trials. And so we've got the tension between trying to get more data in a population that's much more challenging to recruit, and in fact, much more challenging to, in fact, do the studies as well. So that creates, a specific area of, I've said challenge multiple times, but it is a challenge. So I'll stop there, but I think that's where the populations where that approach is are going to be more useful. would be in those younger populations.
Thanks. Thank you, Dr. Nelson. Dr. Batchelor, I was wondering if you could comment on, the role of modeling and simulation, which populations, which drugs maybe, which classes in your study do you think that this approach might be, or do you think it should be used sort of standardly?
So I guess the question's made of several parts. So I'm really interested in the physiologically based pharmacokinetic modeling, and particularly the aspects that bring together the formulation structure as well as the patient. So a lot of the modeling and simulation has previously derived from, models based on liver and distribution of drugs. So the absorption of drugs, the oral absorption of small molecules is getting there in terms of understanding for adults. It's not as refined for children, and that's a little bit due to the limitations on the data that's available. So really understanding the differences and the subtle differences in physiology and the ethics of doing trials to generate that data from children. And then really understanding the formulations and how they interact. So for things like poorly soluble, even small molecules, when we're using technologies, nanotechnology and lipidic formulations, the models aren't as good there as for an immediate release traditional product. Then if you're going to biologicals, again, you've got another layer of complexity in terms of the product performance as well as the patient. And to pick up on what Skip was saying, really, the models are better for the older children, those that are closer to an adult, than the very youngest ones, which is where the models are most needed. So I think modeling and simulation is absolutely essential, because I think we can learn a lot from when the models work, and we can probably learn even more from when the models don't work. And I'm holding my hands up and saying that as an academic is more joyous to us than it would be for anyone working in industry. So we enjoy it and things don't work because it's a question. And I know that that's probably different to a lot of the other people on the panel here, but I think we need to share those experiences, and sometimes the sum of the parts could be bigger than the individuals. At the moment, there's so much work going on into machine learning and big data. I kind of feel if we could integrate so much and get access to that, obviously whilst retaining the IP, if there's any way of sharing that data to really tease out the instances where the models do and don't work, we might understand more about the benefits and limitations of the models.
Thank you so much, Anna. And this is showing that, the increase, interest and use and acceptance of model-informed drug development, and in particular in pediatric development, because I remember that, some years ago, when, all the discussion started in Europe, and of course with experience gained, now the focus and of course the topic is being discussed not only within pediatrics, but also more broadly. And this is why, ICH is quite, a good platform for such discussion to identify a useful recommendation. And also this is showing how, it is important to bring at the table all the expertise. So taking the advantage of having Amy with us, she is a well-known and well-recognized pharmacologist and pharmacometrician. So probably, Amy, you would like to add something on this.
Yeah, I think first of all, also I want to thank you for Skip and Hannah to give the really fantastic talk. Not only trying to identify the gaps and also inspire the boundary of how perhaps quantitative approach and modeling and simulations can help in extrapolation, also bridging study and BAB as well. So one of the questions that would like to follow on from early on is, Skip, you mentioned about safety extrapolations, and then how did you see what is the limitation of the safety extrapolations? And in order to do safety extrapolation, what will be the sample size, also considering of the different therapeutic areas, because you outlined asthma is one of them. And the question can also be answered by Hannah as well, because formulation and safety also hand to hands together? How do you see this both linked together when we want to also understand, will the new pediatric formulation also related to safety? Maybe Skip first.
Yeah, I guess I'll start. The extrapolation of safety is not as well developed, I think, conceptually as the extrapolation of efficacy. But we do it all the time. When we look at the adult program and we see the type of safety data coming out of the adult program, we make adjustments to the pediatric program. And we do that all the time, and beginning to understand how, those sorts of extrapolations. But again, so I mentioned oral contraceptives. So for example, those are approved down to the age of menarche, but often without much adolescent data, depending to some extent on the products that are already combined within those and what's already known, let's say, for similar products or the same product in adults at higher doses, et cetera. So bottom line, we are extrapolating safety when we do that. I think it's difficult in my mind to generalize across programs because you've got both disease-related issues, you've got on-target, off-target effects. Certainly in biologics, you can have the same on-target and very different off-target depending upon the way that biologic has been put together. So I'm a little hesitant to sort of give, here's the recipe, if you will. And I guess one other comment, and then I'd be curious to hear Lynn's take on this as well, that one thing that I think is important in the ICH E11A working group draft guidance is that we sort of open the door to that conversation, but not in a way that says, well, here's exactly what you've got to do. Because I think we need to explore that, we need to develop that, we need to sort of create, if you will, the science of the extrapolation of safety that is not as well developed as efficacy from that point of view. So I guess maybe Lynn and then over to Hannah, I guess, to comment on the work of the ICH group.
I'll take a moderator's prerogative maybe and respond to Skip a little bit, and because I do think that it's a really important question, and I think that the audience is probably interested. I'm, fully in agreement with you, Skip, about the idea that it's not well developed. But I think that just because it's not well developed doesn't mean that we shouldn't be trying to develop it. And in fact, I think what we're trying to move towards, exactly to your point, Skip, is that we have had standards for a long time. And I really think, Dr. Batchelor, your talk was really excellent in setting the stage for, should we be questioning conventional wisdom and dogma, right? And, in the safety world, we've heard all the time about, the rule of three, or, 100 patients, randomized to get a 3%, to identify adverse events at 3% or greater. That's sort of a standard approach. But what I think Skip is trying to point out, and what we hope to do, at least to some degree in the ICH E11A guideline, is to say, look, if we know already about some of the safety, then the idea that you need 100, randomized to give, this is already out the window, because we have some data that we can leverage and rely on. But then we really do have to think carefully about how we create a safety program that will answer questions that we think are important to answer. And in that, how do we justify the number, the length of exposure, whether or not we need controlled safety or not, and whether or not we need, for example, longer term. So all of these questions are open. And I think the goal is, in an extrapolation plan, or an approach rather, in your concept and in your plan, that you should be justifying how much safety data you need, separate from the strict question of how much I need for whatever extrapolation of efficacy approach is needed. So that sort of, I don't know,
And just
medium response.
maybe to add just one thing as well. You know, the complexity is there. So for example, do you have a product where you're going to be administering it across the age range to where you need to worry, for example, about the transition from, you know, the pubertal transition, for example, in growth and development? What have you learned, for example, out of your reproductive toxicology or your you know, studies that have taken non-clinical data throughout that transition in an appropriate animal? So there's all of those questions are fundamentally driven by how similar your model is to your, you know, that in fact is your reference population in that extent to your target. So bottom line, this is a complex area, but it's sort of been a dirty little secret that this has been going on all the time, and we just now are saying, let's talk about it.
Yeah, I fully agree with both Skip and Lynn, your perspective, and also really want to highlight what Lynn mentioned about we need to really have the right trial to collect the nice safety data as a preparation before we can actually analyze how much safety data we actually require for successful extrapolation, and model and simulation do play a role in that. And I wonder whether, Hannah, you want to comment a little bit also on about the safety perspective with formulation?
Yeah, I'm trying to sort of, so a lot of the time the work that we're doing is looking at mapping exposure onto adults and justifying that. The only thing that I was thinking that was tying into this that's really a hot topic at the moment in formulation is this reference person. And a lot of the work that we do is the other changes that happen with age is diet and co-administration with food and all of those aspects, which is our current hot topic in formulation. So if we're dosing children with soft food, then how do we represent that in an adult? So if we're going to give a baby a teaspoon, do we give an adult a tablespoon or several tablespoons? To how do we make an adult physiology more like a child? Particularly for me, I'm interested in the gastrointestinal functionality, but we talk about changes in things like puberty, about sort of some of our physiological changes, but our diet changes going from the sort of weaning phase. So your diet is changing hugely, and we talk about this FDA breakfast as a worst case scenario. But and I know it's not representative. It's not representative of an adult, but it's certainly not representative of like you say a breastfed neonate, where it's a huge high fat diet. But does it have the same effects on the motility? So I think sometimes it's that really. It sounds simple, but it's a really complicated issue to look at how we get that, and we know that food can change the pharmacokinetics and hence the profile of a drug, but that's something that I think we've got to look at, either making a model that represents a child or somehow giving adults a reference position where they're more childlike internally, but I don't know how we do it.
Yeah, indeed, you really highlight a really potential challenge of food interaction study to be carried out at especially very young age. And also there's so many to consider, that the potential of the differences, and also especially for combination drugs as well, which is quite common. So thank you so much.
Can I ask a follow-on question, Amy, to Dr. Batchelor? I'm curious, and maybe the audience knows this, I actually don't, but what is the FDA breakfast? Because my FDA breakfast is usually just a cup of coffee. So I'm curious what the FDA breakfast is.
I don't know that I can remember it verbatim at this point, but it's a high fat heavy breakfast. It's got bacon, butter, and it's not meant to be a typical person's breakfast, but it is meant to cause
breakfast.
It's meant to cause the greatest physiological response. So it's a high fat, high density meal. So it slows GI transit to really change how a product might move through the body. But it's not something you'd want to eat on a regular basis. I think the coffee might be better.
Yeah, which is a bit challenging to imagine a neonates were having a full British breakfast. Okay, so great. And then Solange also, and Lynn, I also see there's quite a few questions coming in live. Should I ask one?
Probably the question, which is may be for Hannah,
Yep.
and which is around, you know, the sample size, which could be selected, justified for relative bioavailability formulation testing for pediatric formulation conducted in adults.
So the sample size, again, it relates back to the standard bioequivalence guidances that are used for adults when you're bridging adult formulations. So I think it's normally a minimum of 12, and most studies that I read about are 12 to 20 people, but I'm just looking to see if anyone's going to disagree with that. So it's quite a small sample size, and where possible, then in healthy volunteers again.
Yep, and then follow on with the healthy volunteer subjects. There is a question coming in to consider biologics. Maybe both of you can answer. Consider biologic if it's subcutaneous formulation development, which sometimes very difficult in younger pediatric populations. How do you see the value of using healthy volunteers to help, again, to inform what would happen in children? Because that would relate, I guess from the person who questions, it would relate it on the subcutaneous depot site differences between adult healthy volunteer to different body composition in children.
I can answer very briefly, but precede my answer with I'm much more confident talking about oral formulations rather than injectables, but I've been involved in a group who are looking at the use of injectable formulations and non-oral biopharmaceutics in children. So I think previously it's been thought that if it's not oral, everything's almost the same, but there are definite changes, particularly in skin composition for younger children. I think, you know, you can see it in toddlers. They've got a much more fatty skin layer, and then they skinny up as they grow taller when they're about 4 years old or so. So there's definitely changes in skin. It's something that I'm active in a group working on at the moment to better understand, but it's not something I could answer right now. Typically, wherever any sort of study can be done in adults and in healthy volunteers, it would be, rather than in children. That's sort of a generic answer for anything pediatric, really.
Yeah. So thank you for that. And then maybe when you can let us know, we can post some reference on the website. And Solange.
There's also a question that was raised by Italo Pogesi. And Skip, you alluded to this topic, you know, when you presented. So it's all about, can we extrapolate based on the results from other compounds in the same indication?
So yeah, I'm looking at some of the questions around safety, and maybe just to expand a little bit more. So, you know, I think it would be difficult. You know, we generally, well, as people know, if you don't have controlled safety data, some safety data might be controlled to where you can begin to tease apart, you know, the drug versus the disease. And if you have coming out of adults, let's say placebo-controlled safety data, you can do that. Many pediatric trials are not placebo-controlled because we're extrapolating efficacy. So we want to do an exposure response type of approach, or we might use an external control of some kind. So in pediatrics, then we will not have that kind of controlled data. So we are sort of relying at least on the adult data to have some understanding of that disease drug difference, if you will. To go between then classes of drugs, I think, you know, would be a challenge. To stay within that, I mean, arguably one could think of certain closely related indications. I think a good example is psoriasis, psoriatic arthritis, where in some sense, I mean, those are so closely related that I think we'd be willing to take the safety data, both adults to pediatrics, but also from psoriasis into psoriatic arthritis, for the same drug. As we begin to sort of move into different drugs, I think, you'd really have to look and see what's the difference with adults as well. So it's between drugs of different classes, I think would be much more of a challenge than within drugs of the same class or across indications exposed to the same drug. But that's, again, these are very general comments, and one really does have to get into the weeds of the specific disease and what's going on. I might ask, there was also the right study, made this downsize the role of ROW. I don't know what ROW means. I'm looking at Italo Pogesi's question. Do people know what ROW? I mean, that's an abbreviation I'm not familiar with.
I'd say rest of world, but I'm not sure.
Maybe Italo, you could put that.
Real world evidence, what Italo meant, I think. Yep, the ROW he mentioned is real world
it was a
evidence.
typo then.
Yeah.
Okay,
Yes,
that's fine. I think, this is a very interesting question. I mean, to the extent that we begin to use extrapolation more robustly and appropriately in the efficacy domain, by definition, as I pointed out, the safety database gets smaller.
Yeah.
And so I would argue that to the extent that we are extrapolating efficacy, one could say there would be a more robust role potentially for real world evidence. Now there's a tension there because FDA and EMA label it as, quote, safe and effective. Although we know that that means safe enough. It's not as if it's absolutely safe. So one interesting question would be the extent to which we try to develop more robust mechanisms for the use of real world evidence for safety in the context of a more robust use of extrapolation.
Yeah, so definitely, I think the increased use of real world evidence will become a trend, and we do have a section also tomorrow to discuss further on the definition and different real world evidence. And follow up on Italo, I think what you mentioned, Skip, was really interesting about the differences between extrapolate of similar disease, compound treated similar disease, or with the same mode of actions, because there's a lot of the time when compounds with the similar mode of actions, people do expect to have similar efficacy, at least when they were evaluated with the competitor, and also safety profile. But would you agree that that would be more encourage people to actually comparing across different similar mode of actions, classes of drug, and then actually start to develop database to evaluate that?
So Amy, let me be clear on the question. So the extent to which we can extrapolate based on mechanism as opposed to
like which way, what is the pros and cons, then it will be by continuous learning and confirming that we will have to put like a database to look at different classes of drugs, that how is the safety and efficacy profile similar,
right, and
then to work on the next part of the framework, perhaps.
Sure. I mean, that could be done. I think that certainly my impression is, and I'd be curious for Lynn's thoughts on this one, is that there's more challenges certainly in extrapolating when, let's say, you're first in class, as opposed to second, third, or fourth. Now, I would arguably say that if, let's imagine you're coming in with a first-in-class product into an indication where there's already been extrapolation that's been accepted, the disease similarity is pretty established. That really where the uncertainty is that response to treatment similarity, and so some amount of extrapolation would be appropriate, but whether or not you'd be willing to go to the same degree as you would for a compound that's had for a class that has a track record I think is an open question.
Yeah, Skip, I would agree with you. And just very quickly, one of, there are many examples, but the one I'll throw out there is in the division of addiction and analgesia, and there's another, I forget the new acronyms in our reorganization, but we convened a workshop just a couple of weeks ago, and we accept extrapolation as a standard policy, and that means that we agree to match exposure, to evaluate doses that would match exposures. But that's limited to acetaminophen, NSAIDs, and opioids. So if there's a mechanism of action that's novel, to your point exactly, Skip, disease process we feel is the same, at least down to 2. But if the mechanism of action is different, then the response to treatment may be different. And so there's not sort of a standard acceptable policy about extrapolation if it's a different mechanism of action. That's just one example. I'll take the liberty to ask the next question because I know the questions continue to come in fast and furious here. I saw another, I thought I saw another question about rare diseases. I don't know where that went. Did that get removed?
I was answering that. I can say that it was someone asking about the ethics of including children in rare diseases, and the comment that I wrote in answer to that question, that there's a session tomorrow on patient experience and engagement. So I was just going to redirect the person who asked the question to, I think it's Mark Turner talking tomorrow, my evening, your lunchtime. So I was pushing them to that.
And thank you. No, thank you, Dr. Batchelor, because that's a really great way to, again, remind folks to stay tuned tomorrow, because we're going to have a very important discussion about how we engage patients and the patient's perspective in enrollment in clinical trials, particularly pediatric trials. I just want to make sure we're catching all the questions. Amy, Solange, do you think we've gone through all the audience questions? Because we have a few others that I think we might want to ask as well.
Yeah, I think we do have time to ask, go through all the questions. There is one question on IND. Maybe that one also good to ask, Lynn and Solange.
Here it is. So the question comes through, what situations or criteria for the industry would it be necessary to file an IND amendment or a new IND for a pediatric formulation? And I guess I'm supposed to be the expert here, and I don't know the answer to that question. So I would have to refer to my regulatory colleagues in the divisions to answer that one. I don't know if there are others who can answer.
Well, Lynn, let me take a step. I mean, generally, an IND is indication and drug specific. And as you know, the pediatric protocol that's submitted, follow up as an adult protocol for PREA or BPCA is submitted to the same IND as the adult program. So I have never submitted an amendment for a formulation, but I would suspect it would go to the IND that is that disease and indication.
Yeah, that's my recollection as well, Skip. Yeah, but of course, I mean, Lynn, if there's the opportunity, I mean, to have, later some answer, that would be helpful. Definitely.
Certainly.
Yeah.
I mean, on the ethics question, I think one thing that I found interesting about Hannah's presentation, I mean, part of the reason I think the regulatory agencies have generally said that you should do your bioequivalence studies in adults is ethical concerns about, you can't really take healthy children and put them in a study where you're giving them active compound. That would not be considered appropriate. And then the question is, well, how do you design a study in children who could potentially benefit from that drug if you don't quite know? So I'd be curious to hear Hannah reflect on what she thinks ought to be done to make a more nuanced approach, given the data that she's presented, which I think is very provocative, in the context of the clear direction from the health authorities that these should be done in adults. I'm just curious, what would you suggest we do differently?
That's the magic question. If we had an answer, I think we would be doing it right now. I think we've really got to understand the mechanisms behind the differences we're seeing in children and adults from a physiological point of view. I think if we have the right information, we could build the right models that would de-risk this, and we would know which compounds were subject to risk. So from the work that we did, we were highlighting it was very much poorly soluble compounds. And I'm talking small molecule here, and I have to apologize for it being so limited. So I think we really need to understand what happens in the dose adjustments. When we're doing the studies in adults, it's really tricky to understand what the effect is of the dose. Oh, are we testing the pediatric dose in an adult, or are we dose normalizing for the size of that patient? If we're co-administering with food, what foods and at what volume, and how are we really trying to make sure that what we're doing is extrapolatable to children? I'm not sure if that's a word. So in terms of what do we do, I think there's still a lot of work that we need to understand what the major risks are, and then design models that are mechanistic that understand those risks. I don't think we can do a one size fits all, but there is not a magic answer right now, I'm afraid to say.
Yeah, I mean, where I see this as a challenge is my impression is there's an increasing desire to go direct to phase 3. In other words, you've got your, in the extrapolation context, you've got your adult efficacy data, your proof of concept, you've established that. And so instead of starting with a PK, because you then lose that number of patients to put into your phase 3 trial, let's say it's not just exposure matching, exposure response, et cetera. But if you get that dose wrong and you miss the exposure, you know, you've basically wasted however many patients it took you to kind of learn in the process of sorting out what the right dose ought to be. So it's the quality of the models is a very important component of being able to go to that approach of trying to do only one trial as a direct to phase 3 approach. So
Yeah.
So
very important work.
clearly a complex issue, and I'm sure, Lynn, you remember at the time of the revision of the E11 guideline, at that time it was said, oh, we need to investigate further what, you know, could be recommendation for pediatric extrapolation. And the other topic that, you know, was suggested to ICH was pediatric formulation. And we are currently, of course, working on the 11A pediatric extrapolation guideline, a work that you are driving, and of course with colleagues like you, Skip, and others. And so maybe this could be, you know, the idea to let's say, look into this pediatric formulation a little bit more, knowing that since then experience has been gained, and listening to Dr. Batchelor, well, clearly there's a lot, you know, to be said, a lot to be recommended, and still, you know, the need to have experts discussing together to address remaining uncertainties, which will be quite helpful for pediatric development. Yeah. What do you think, Lynn, of this proposal?
Well, I couldn't agree with you more, Solange. And again, as you point out, it was well recognized at the time that the E11R1 was undergoing revision, that there was a need to be more, to consider a more granular guidance on pediatric formulation. So if anything, I think Dr. Batchelor's work has highlighted the need for, you know, further considerations in that area.
Thank you, Lynn. And I think that there's one last question, and I think it will be probably the last, you know, the one that we will be able to address. So this is one raised by Georgi Zorini. So another question, whether the established safety profile of a drug with adult indication could be 100% extrapolated to the pediatric population in different indication, even if for some of the listed adverse reaction in the label. No single AE was reported in the pediatric trials.
Maybe I can start with a couple comments. So let me first answer that for the same indication, as opposed to a different indication. You know, the pediatric trials are often much smaller. So let's imagine you use exposure response, or even if you want to, I mean, for example, the lupus example, there was 93 kids total, of which only, if I recall, 53, I'd have to look at my slides, I think were exposed to active product. So, you know, the rule of three, if you have 50 kids and you see nothing, I mean, the confidence interval for that is quite broad. And so by definition, you are extrapolating the adult safety to some extent when you say, we didn't see anything different. But you have to recognize that the confidence interval around a 0 observation and only 50 episodes, I don't have the statistics right with me, but I suspect it would be up around 10 or 15% potentially that you would have missed that. So you're really only going to pick out common and to some extent perhaps dramatic adverse events with that size of a population. So you are extrapolating efficacy. I mean, and then you're relying on post-marketing safety to pick up something that you might have missed. To do that for a different indication is, you know, again, some you can, psoriasis, psoriatic arthritis, I've mentioned. Others, you know, would be more problematic. So I think, but that same conceptual challenge of seeing no events in the pediatric trial, leaving a fairly high confidence interval around that, you are potentially extrapolating. But I think the issue there would be, can you do it for that indication? I wouldn't take what I just said and apply it to just different indications across the board. So I don't know if there's, you know, Lynn, if you want to add anything to that or not.
The only thing I'll say is that to add is that not only is it problematic because, you know, the pediatric safety database will necessarily be smaller in many cases, but it's often the case, and this is just the reality of safety evaluation, that the adult safety is not completely understood or known at the time of the adult approval, right? So even if we have, you know, adult controlled safety to rely upon, you know, there are often still unanswered safety in the adult population, which, you know, carries over to the pediatric program as well. So it is, it's a, as you say, Skip, it's complicated. So I think what we're looking for, though, in the end is a justification and a rationale and a review of the data that you have to support what we think is, what you think is needed, or what we think ultimately will be needed in the pediatric program.
Thank you so much, Lynn. I think that it's time now to thanks the panel and also the audience for their very interesting question, showing us, as you know, the E11A expert work group, that, you know, the work we are doing is quite useful, and that we expect that at the time the draft guideline will be released for public consultation. It will meet your expectation, and of course your comment will be very welcome. So I'd like to thank my colleagues, moderator, and also the fantastic speakers, expert. We've had the opportunity to listen to today, to hear, you know, their very relevant answers, showing that it's all about, you know, the bringing in the discussion the right expert, so that, you know, working together can be, you know, for the better, for the best in pediatric drug development. So with that said, Lynn, any particular concluding remark, please?
No, you said it all, Solange. Thank you.
Excellent. Thank you so much, Lynn. Thanks, I mean, to have, joined us, knowing that initially, as you remember, we were expecting Dr. Mulugeta to join us, but unfortunately, for personal reason, she had, to cancel attendance, and we are really grateful that, you proposed to join us. So clearly, thanks so much. And it was a real pleasure. So lots of questions showing that there's lots of interest, and that, I'm sure that in the coming months, years, there will be even more experience gained and more shared learnings, knowing that later today and tomorrow, there will be also a very good discussion, very good topics to be discussed, and that are worth considering using for pediatric drug development. So thank you so much.